So thank you for having me. I know I'm the last talk. I feel no need to get through all this. If it just turns into us arguing with each other, I'm happy, or discussing something. So please feel free to interrupt. You've seen me interrupt the whole time. So I know this is a very mixed audience. So for the experts, I apologize for the introduction, but I always find that in these mixed audiences, it's much better to have a longer introduction than it is to delve into details. So I'm going to tell you about some thoughts about how we're thinking about biological data in the modern world where AI and machine learning is everywhere. And as I said, there's going to be a reasonably long intro. And then I'm going to show you two cases where I think, these modern, and by AI, I just mean transformer-based architectures.
Just technically, is your mic is very loud for me.
Oh, sorry. I don't know. Is it on? Oh, I think I'm just very loud. I'll talk softer.
You go ahead.
Is it on? I think it's only for Zoom.
If it's not,
Oh,
make a difference. It's just
Oh.
if it's just your loud voice, you
then I will stop yelling, which is my I get made fun of by my kids because I butt dial them when I'm teaching and they're like, your teaching voice was so annoying. You just scream at your students, which is roughly right. Okay, I'll talk softer. I don't know how loud it is. I can never tell. Anyway, so I'm going to tell you about how transformer-based architectures. There's this idea that you can just throw them at any problem in biology on any data set if there's plenty of it, and some magic will happen. So I'll try to tell you through practice when we found that these kind of ideas work, and I'll show you when I think they kind of pretty spectacularly fail, despite claims from many people to the otherwise. And then if time it permits, which it probably won't, since this is a math thing, I wanted to tell you about how maybe using human AI can tell you some interesting ways to analyze high dimensional data that I think even today, the LLMs, even in swarms, probably don't have such a good shot at doing. But as I said, and a lot of the framing of this talk is this rant I put on the archive a year ago because the AI hype was I was being told over and over again that we should stop being theoretical biophysicists, stop thinking about statistical physics because we were going to be put out of business. And I'm going to touch on many of the themes, especially in the morning talks about biology. And in particular, what makes biology so complicated is this idea that you have some genomes where we have a lot of data, some DNA on different sequences. You have an environment, and you have this very complicated process this is the typical biological diagram where you have lots of words and lots of arrows between them, which is supposed to say there's some complicated stuff that happens. And depending on what your genome is and what the environment is, you get either a tall plant or a short plant, right? And it's indeed a very complicated process. And the thought has been that what's amazing is that if we measure more and more of these arrows, more and more of these components, then we'll make a lot of progress on understanding how this happens. And indeed, the central, there's many parts to this whole thing. So there's, I'm just going to tell you about the biology. I'm going to talk about one place where there really has been a lot of progress is in central dogma, where you have DNA, which turns into RNA, which turns into an amino acid sequence, which turns into a protein 3D structure. And what's nice about this is fairly conserved across organisms. You can take a protein sequence from one organism, put it in E. coli, and with, I would say, reasonably good probability, if it's not too complicated a protein, it'll fold into some very similar structure. So this is, this is fairly conserved across organisms and largely context independent for most proteins. But, a lot of other stuff like cellular identity, where these large transformer-based architectures are being applied, this is far from true, right? And so the kind of question in biology people are now interested in, where there's other large data sets being generated, are things like development, where we know that every cell in our body has the same DNA, but through some complicated process where genes are regulated, certain proteins are expressed in certain cell types and other proteins aren't expressed in other cell types, you kind of get this complicated process where you get that is controlled by another bag of arrows that we're going to try to replace with a large language model. You get some complicated emergence of many, many different cell types, right? But what I want to point out is this is like fundamentally, in some sense, a very different kind of biology from the central dogma from DNA to proteins. And I'm going to try to emphasize that because that's going to be very important for what follows, right? And so what's interesting is, of course, this is a very complicated process. And the thing that's happened in the last 20 years is that biology has gone from data poor to a data rich science. And the main reason for this, I would say one of the main reasons for this is the sequencing revolution, which is that we can measure, DNA sequences and turn many other biological problems cleverly, as you heard in the first program, into problems of measuring DNA sequences. And from measuring these sequences, we can compile lots and lots of data. And the amount of data is quite staggering. So, you go from something like, you have 10 to the 10th bases of DNA in an archive in 2008 to 10 to the 15th basis, in 2023. And sequencing costs, drop for a genome from, something like $1,200 to under a cent, all in the last 20 years. So you can imagine the amount of data that's being produced now because, these 5-fold, 7-fold drop in costs combined with robotics, as you heard about. So, I would say the crowning success of machine learning methods in biology has been on this problem from DNA sequences to protein structures. There were 2 Nobel Prizes given out last year about this. And I think what underlies it all is really this amazing astronomical growth in databases. So you have, if you see here, the units of red and blue are very different. So you have, millions of sequences. So you have 300, this is whatever, 300 million sequences. And you have something like, something like 158 million protein structures. And what's nice about this kind of data is that it's very, very clean. So one of the things that we haven't talked so much about, even though it's a big data question, is about how good is the data? Is it very high quality data? What is the nature of the data? And I think if you don't take away anything from this talk, you should take away that all data is not equally, like the quality of data still really, really very much matters. And so what's nice about this particular problem is that both DNA sequence data and protein structure data is extremely quantitative and extremely clean, right? So you have these 2 giant databases of very, very clean data that talks about going from coding sequence to protein. And on the other hand, what's happening now more lately is this bigger mess of cellular biology. The data is less clean. So you're trying to measure things like the number of proteins, the amount of mRNA that's made, the transcription factor that's going on. You're making atlases of gene expression. And this is, this is a bunch of technologies that you have. No matter what technology you have, the interesting thing about it is that with all these technologies, what you can do is you can measure the expression of, it's called single-cell RNA-seq, but it's essentially a proxy for what proteins are being expressed in a single cell. And so you can measure, make basically a measurement of how much each protein is being expressed in a cell. It's really mRNA, but it's supposed to be a proxy for protein to some degree, right? And what's interesting, of course, now is that the same explosion you saw with DNA sequence data, you see the same explosion and drop in cost in this single-cell RNA-seq data. So, you went from something like sequencing a cell cost about $1,000 in 2010, and now it costs about a cent in 2025, right? And the number of sequences go from 0 to 10 to the 6. So you just have a lot of data. And people are building a lot of atlases, and so you're measuring all these kind of things that a new kind of biological data, which is basically data that's supposed to tell you about how these gene regulatory networks that give rise to cell identity work. And of course, the big thing that people are making are atlases. So atlases are essentially a name for what biologists call databases, and we'd like to make sense of all this stuff, right? And so I think this is only going to get more pronounced. What's going on is that people are measuring more and more, complicated biology. They're measuring all of this at scale because all these things can be turned into sequencing problems. And then what's supposed to happen is there's some machine. It's unspecified in this review from 2 years ago, but there's like some funnel. And out of the funnel comes biology, right? Biological insights. And of course, it's unclear what that machine's going to be, but because of the spirit of the times, and so I think what's interesting is I think Sidney Brenner, who passed away recently, already in 2012, a decade ago, 13 years ago, was kind of warning us about this situation, which is that, he has this quote that I really love. It's in a retrospective, celebrating Turing. He says, technology gives us the tools to analyze organisms at all scales, but we're drowning in a sea of data and thirsting for some theoretical framework with which to understand it. And I would say that very much characterizes the state of biology. We're very data rich. And we've made lots of progress. I don't want to give you the impression that there's no progress being made. There's, of course, immense progress being made. But I would say, on a whole, in subfield after subfield in biology, there's this feeling that we have more data than we know how to synthesize and organize. And there's lots of technical challenges. Once you move away from DNA sequence data and protein structure data, the data is often extremely noisy. It's extremely sparse. And I think another thing that's underappreciated about it is it's quite continuous. It's not discrete like base pair data. And so, for example, if you look at this single-cell RNA-seq data, which is going to play a big role in the second half of this talk, if you go look, so what you're going to get is you're going to get data like something like this. Each column is a different cell. And you're going to measure how many mRNA transcripts there are in a cell. But what it turns out to be true is that 80% of the measurements turn out to be zero. So if you look at a given cell, any given cell is not that informative because these processes are very noisy. Though you can do these things at scale, the kind of data that's being generated is much more noisy than any DNA sequence data that most people are used to thinking about. There's also missing data, and it's hard to measure stuff. It's not really important. And there's also very important conceptual challenges, which is that the environment is almost entirely ignored. And the second thing is that there's the biological problem of context, which is that the same element in biology, depending on what context you put it in, behaves extremely differently, right? So people have all these problems in mind, and they want to do something. And so, we could come back to Sidney Brenner's warning, and, so people are trying to think about what to do, and the field's answer to this challenge is, of course, that AI will save us, right? And this is not just something I'm saying. This is a retrospective from maybe last year in Cell, where about 50 leading people told us that what we're going to do is we're going to take all this data, we're going to build a lot of AI foundation models, whatever those are, and then from the foundation models, we're going to be able to do all kinds of biology. So this seems to be the prominent program of the field. And of course, it's inspired by the incredible success of AlphaFold and and Rosetta and things like that. So it's not based in nothing. But the question I want to talk about is I think we have to have a slightly more critical view towards such a program, because all data is not created equal. All biology problems are not created equal. So I'm going to start with where I think this kind of approach has some chance for working. And then the second part of the thing, I'm going to show you another very large language model that was trained that we find we can do better with 5 minutes on a laptop than a model that was trained on 1,000 GPUs, 1,800 GPUs over a month. And so I think it's just a lesson in, I think one has to be critical about what these things can do and not do. And often the devil is in the details of the data and the details of the problem. And we can't, it's hard for me to believe there's gonna be a one size fits all to all this stuff. So, as I said, I think if you go read the news, it's rather surprising what's going on. So, for example, Steve Quake, who is a person I respect immensely, gives me lots of money, says stuff. You can find him saying stuff like he runs the CZI, the Chan Zuckerberg Initiative, and you can find him saying stuff like, "Our goal is to create a computational tool so that cell biology goes from being 90% experimental and 10% computational to the other way around." So they have this whole science. The model I'm going to tell you in the second half, in the second part of this talk, is the first model that's supposed to be part of this virtual cell project, which is called the TranscriptFormer. Then you have people like Dario, who tell us that they're gonna, accelerate 100 years of biology into 5 or 10 years, and then uses an example of CRISPR where he basically misrepresents the whole history of how CRISPR was discovered in his blog post. So the question is, on some level, I'm a big believer in these tools. On the other hand, I feel some obligation to get up here and say some of us who believe in the tools but understand their limitations, it's our job to point out when the hype becomes out of control. And so I don't want this to be a negative talk. I'm just telling you, I feel some obligation to point out that sometimes these things are applied uncritically, and then tell you why I think, when I think these can be applied and when these things can't be applied. So the reasons for optimism are obviously this, the automating the sequence to structure prediction, which everyone understands, but of course, the prerequisite for that was extremely high quality, low noise data, right? And, the amazing results of AlphaFold 2, everyone, I won't go over it, but of course, it's rather amazing. And what you might know less well about, which is the inspiration for a lot of stuff in biology, are actually these protein language models. So protein language models literally were, for those of you who are experts, people took the BERT architecture, which was a very early pre-chat GPT architecture, and what people did was they took a bunch of sequences for proteins, and they basically trained these language models where the goal was you take some so a protein is defined by a string of amino acids, which are these letters here, and the goal was that given some of the amino acids, the idea was to just predict the other amino acids that were there. And this could be done either in a bidirectional manner or an autoregressive manner, or it could be done in a masked sequential manner. It doesn't seem to make much difference how you do it. But the interesting thing is that you basically learn a generative, conditional generative model for how protein sequences are generated. And what was interesting about these protein language models, and so one that a prominent one that was trained by the Facebook team was this thing called ESM2. What was interesting about these things is that you could take these large language models where the tokens were amino acids, and so you take an amino acid, it gets mapped to a vector, you get an output vector, right? And these amino acid embeddings, and what was interesting is you could take these amino acid embeddings, these embeddings of these proteins, so you've taken proteins, you've embedded them in a meaningful vector space. What turned out is that these vector spaces, just like the Python programs we heard about in the last talk, seem to have a lot of structure, and you could use that as inputs into downstream tasks, and these embeddings basically improved all these, improved the performance on lots of stuff. So you could figure out how to make another protein, predict whether another protein binds, try to fine tune what the structure exactly is right. So there's these incredible idea that and the protein language models, even more than AlphaGo, that you can take things like protein sequences or the family of all protein sequences and meaningfully embed it in a vector space. And this vector space then could be used for downstream tasks, right? And so these things are quite large, 15 billion parameters, 65 million sequences, and the attention windows are, 2,000 amino acids, so quite big, large proteins. This has really inspired a lot of work in the field. So, and the lessons of these protein language models I would take away is that the language of proteins can be captured using this transformer-based architecture. So it's kind of surprising that the sequence to structure, relationship is well captured just by a generative model for how the amino acids are generated, right? I've never told you anything about the structure. I only do one-sided training on these things. And they seem to be great at dealing with the problem of context. The same amino acid in a different sequence, of course, behaves very differently from, if I take the same amino acid and put it in two different sequences, it'll behave very differently in the three-dimensional structure that you see. And so you get meaningful representations of amino acids and proteins in a vector space. And just like with natural language, scale matters. More sequences, more parameters, better performance. And the last thing that's very interesting is that these LLMs seem to be really great at learning demographic and evolutionary relationships in these DNA sequences data. So the reason these sequence to structured models work so well is because we know there was an evolutionary history through how these proteins were generated. And that process of how these things were generated through evolution and through mixing of populations, as we heard about, that generative model seems to be very well captured by these LLM-based things. Yeah, go ahead.
You do AlphaFold like predictions using language?
Yeah, so what you usually do is you take these embeddings, and what people usually do is they actually just take a piece of the AlphaFold folding software. Like this folding trunk is actually from, I don't know what version of AlphaFold, probably AlphaFold 2. And they take the vectors as an input into it, and they train it. So you can't do it off the shelf. The idea is that you take these vector embeddings at the top, and then you train specialized models to these downstream tasks. But this is essentially a good kernel. The learned kernel of these language models basically map them to a space that seems to have meaningful dot products and meaningful similarity scores. And so that's basically the way these protein language models are used. They're used as a first layer of encoding sequences into a meaningful vector space that can be fed down to various downstream tasks. And the interesting thing is that this same encoding, which is just based on auto-regressive sequence generation, seems to work for many downstream tasks. Which is surprising. Yeah, go ahead.
Do you have any guesses as to why that's true? I agree that that's kind of weird.
Yeah, so our current working hypothesis is that it's because, well, I mean, it's the same reason machine learning always works. Data is not random. But I would say in this particular case, I think the process of evolution, which is the process of duplication and divergence and mixing, is something that these kind of large language models are particularly well at capturing. And if you think about how languages evolve and words evolve, it's, we heard of essentially an evolutionary language talk. I think the isomorphism is quite close, I would say. I think those are the kind of relationships they seem to be quite good at capturing. I don't know, maybe other people have opinions.
I mean, it's not entirely clear whether they're capturing evolutionary data constraints or there's biophysical constraints that proteins have to satisfy, which are global, and that they'll learn those constraints. I mean, is it obvious that they're learning something about phylogeny?
It's pretty clear they're learning something about phylogeny, not from the ESM calculation, but there's architectures where you can take where you train, like MSA transformer. This is getting kind of technical, but you can train not on single sequences, but you can train on multi-sequence alignments. And then you can have attention heads across the sequences, and then you can have attention heads across the MSA, across the alignments. And it's clear that the attention windows of the alignments across the alignments actually capture quite well evolutionary relationships. So
Train using aligned sequences.
yeah, but I don't think there seems to be too much difference in what you can do downstream in them. So I think just the generative process, because it's evolutionary, tells you about evolutionary constraints. I mean, that's my working. I don't think the short answer is no one knows. I suspect that to be true. I mean,
Let's come back and discuss it.
Okay, sure. All right. So, I walked into Michael's office with Krishna, who's sitting there, and we wanted to do a stochastic process problem about in pop genetics, and Michael talked us into doing a machine learning project instead. So I'm going to tell you about a problem we heard about in the morning with Michael, and then we called Gautam from Princeton because Gautam is our LLM expert and also thinks about genotype to phenotype maps. But one of the first places we wanted to think about this was in learning these genotype to phenotype maps, and we're going to exploit the same kind of experiments you heard in the morning. And the reason we think this is probably an ideal scenario for thinking about this is that there's high quality quantitative DNA sequence data that the way these genotypes are generated is through a similar evolutionary and demographic process. And in particular, we have there's often high quality phenotypic measurements for quantitative traits. And so we've been exploring, this idea of whether you can do stuff in learning genotype to phenotype maps And the basic idea, of course, is that you have some processes like demography and evolution that give rise to DNA sequences. The genome, along with the environment, gives rise to a phenotype. This is a big black box, and we're going to try to replace this big black box with a transformer, which is and the experiments we're going to work on are very similar to the experiments Gautam described in the morning, where you have two yeast strains. They're actually no, they're not the same exact ones. You cross them, you make many, many genetic backgrounds, then you grow them in a bunch of environments, and you measure their growth rate or their fitness, whatever they're called, through DNA, through some very clever DNA barcoding stuff that Michael and his lab figured out, right? So you can do this thing at scale. You can measure 100, 10 to the fifth, the growth rate of 10 to the fifth things in hundreds of environments. Right? And so this is data on a scale we could never get before. And what I should point out about this data in particular is that it's actually the demography and the evolution of these are very simple. The backgrounds are just generated from a single cross. But we thought this was a good test case for testing this out. And where we expect this transformer stuff to really shine is when you have to deal with population migrations and things like that. All right. So we just wanted to play around with this. And so, just like in protein language models, you instead of inputting, amino acids, what we can do is just like Gautam was talking about, you think of a genome as L loci that vary. In these crosses, just like you had plus or minus one, I have orange and white squares. So every background is some binary vector of length L. I have some environment, and I measure the growth rate in these environments. And so what we're going to do is we're going to use a fairly standard transformer architecture with some things that you don't have to worry about. And the idea is that you can embed each genome, each loci, you can embed the genome. The interesting thing, of course, is that in Michael's experiments is that we can actually learn a token for the environment as well. So this is something that Gautam and I have, there's lots of fights about how we should do this exactly within the group. But you take some genomes, you take an environment, you spit out a growth rate, and you ask what's going on. And what is interesting is that we're interested about is two things. We're interested in demography and how these genomes actually evolved. The second thing we're interested in this thing that Gautam will also refer to, which is epistasis, which is the idea that you have background dependent growth rates. And so you can do stuff with synthetic data where you basically take the actual backgrounds, you make some synthetic growth rates, you can vary the amount of epistasis. So here, this is very high epistasis, very low epistasis, and there's and you can train these things and they work reasonably well. So there's two lessons I think you should take away from this. First is that linear regression is really, really, really good most of the time, unless you're in a very highly epistatic regime. Right. And the second thing that's surprising is even though these models are quite nonlinear, they do quite well on linear data too, for reasons we don't really understand well. You can train these things in various ways, but they work, I would say, remarkably well. And, they do well on experiments again. But they don't do so much better than linear versus pairwise things or anything else, right? So classical statistics, even linear regression is pretty good at capturing these complicated phenotypes, at least in these settings. That's what we find. And part of the tension we don't understand is how to reconcile that with this thing that Gautam sees, which suggests that there should be all kinds of higher order interactions. So there's some very funny tension between statistical inference models that are used for statistical inference and what the reality is. The second general lesson that's fun about all these things is that what these transformer-based architectures seem to be quite good at is transferring knowledge across environments. And so what you can do is you can, train on some subset of environments, have limited data on another environment, and what you can see is that instead of needing something like 10 to the fifth samples, if I have something like 10 or 30, 20 measurements in a new environment, you seem to be able to predict growth rates across this new environment. And this is because there's shared pathways. We know the biology of how things work is that there's shared pathways and the biology is not different in every environment. So, consistent with these protein language models, I would say these architectures are working pretty well in this simple setting. But what we're most excited about is the idea of incorporating the idea that here, the genomic backgrounds were just generated by just crossing two things. But we know the real way this DNA, these backgrounds are generated is through a very complicated process of selection and demography. And so we wanted to understand, what we're working on now is trying to understand when you have this very complicated backgrounds, right? Where you have lots of interactions that are very complicated contextual loci dependent things, do these transformer architectures still work well? And we're doing that using these kind of evolutionary simulation frameworks, which basically, simulate these kind of things that we heard about in the morning. And what we find that's surprisingly good is that now once you start putting in, these kind of very complicated evolutionary and demographic processes for how these sequences are generated, the gap between the transformer models and the linear regression models, or the linear regression and pairwise models, seems to get much, much bigger. So as expected, these kind of transformer-based architectures seem to be quite good at capturing the complex relationships within the genome that are generated by evolutionary processes. And the data here is just, we simulated an out of Africa model with two populations, Africa and Europe. It was generated, we simulated 5 different migrations with the same parameters. Then we put 5 different synthetic, phenotypes for each ones. And what's interesting, of course, is that, regression famously does very badly at predicting phenotypes when you have, when you train a model on, say, Europeans, it's very bad at predicting things on people with African ancestry. And what's interesting is that these transformer models seem to automatically learn these structures, at least in our simulations. And should add, these models are non-trivial to train. They're a little bit big, and the data is big. And Krishna spent a lot of time debugging how this works. But we're pretty excited about this idea, and this idea that we might, and Gautam has been pushing us to think about whether we can get a universal embedding of these genetic loci and train all the phenotypes at the same time. And in this way, maybe we can get a meaningful vector representation of genomes. Because I think if we had meaningful vector representations of genomes, you can imagine there's some more downstream tasks that you could do that we couldn't do now, just like you do with protein language models. So we're pretty excited about this. But it's very close to what a protein language model is. To us, it is just, it's just a different place where the data seems to be good, where the phenotypes are quite quantitative, it's DNA sequence data, and the transformers work well. So with Juan, who's also in the audience, more recently, we've been asking, about the same thing, but with different kinds of data and different questions. So the question we've been asking about is these questions that about cellular identity and gene regulatory networks and single-cell RNA-seq data. So, so just maybe in March, the CZI and Virtual Cell released this, the first of their virtual cell models, as they call it, which is called the TranscriptFormer. And the basic idea now is that they want to have meaningful embeddings of cells. So remember that each cell is defined by the fact that it expresses different proteins. So what makes a muscle cell different from a stomach cell, different from a lung cell, is that different proteins are expressed in each thing. And these single-cell RNA-seq measurements are basically trying to find proxies for what proteins are expressed in what cell. And so the basic idea is you go out, there's huge databases of this is this cell type and this is its expression level, right? So these are these databases. And what's interesting, of course, what they were excited about is that you can do this not just for humans, but you can do this across all kinds of species. So I think the title of the paper was something about 1.5 billion years of evolution or something like that. I don't remember exactly what it's called. And the idea is that you take all this data, you put it through a transformer, and now you get vectors for each gene, and you get a vector for each cell and these embeddings. And these embeddings presumably have lots of information in there, just like the protein language models did, that capture meaningful biology. And so they should excel. If you use these embeddings, you should be able to do lots of downstream tasks about biology in a way that would be much better than if you hadn't used this big transformer to embed all this stuff. Right? So the kind of tasks you might want to do is I have cells in one species. Can I predict the same cell type in another species? Can I, tell you if a cell is diseased or not? Can I, classify, if I give you an arbitrary gene expression profile, can you tell me what cell type it is? These are the kind of expressions they want to do. And just like in the protein language models or in the transformers where the goal is to map biology to meaningful vector spaces, the idea here was that you could take this single cell RNA-seq data and map it to meaningful vector spaces that capture all the complicated biology of these gene regulatory networks. Right, so as I said, the model is quite long, quite big. It's 3.5 trillion tokens processed just to train this model. That would be like 40 hours on 100 H100s or that's right, 4 days on 100 A100. So this is not a slouch of a model. It's a billion parameters, right? It's not quite the most giant model, but it's not something I would have the resources to do it in my cluster or even at Harvard or Princeton or something like that. So just because you're going to see these names, you can ignore all this. They actually trained 3 different versions of these models. They just start with a TF. And the difference between them is how many different species they trained on. So this TF sapiens is just trained on human data. TF exemplar is trained on something from fruit fly to humans or something like that, C. elegans. And TF metazoa has sea urchins and fruit flies and very, very divergent species. But it's just the training data that was used to train these things, right? So you're just going to see these in graphs. So I'm just pointing out that it's actually not one model, it's actually 3 transformer models trained on slightly different kinds of data. So, but there's reasons I think one a priori should have been skeptical, and I will show you that they were well-founded in about a slide or 2. One is that single-cell RNA-seq data is really different from DNA sequence data. I know there's been this idea that more data is always good, but I would say more good data is always good. More noisy data can sometimes just be noise, right? And I think Renaissance excelled at telling the difference between noise and signal. And I don't think large language models solve that problem for you ever. The other thing is that there's systematic errors, and I think another thing that at least I dogmatically believe is that the kind of variation you're trying to capture in protein counts has a very different generative process than the kind of things that happen in evolution and demography, where you have mixing, mutation, fixation. This biology is much more complicated than that once you abstract it away. And so the other thing is that unlike protein structures, gene regulatory networks are extremely context dependent. Because we know even protein languages and AlphaFold do much worse when you have things like chaperones or you have intrinsically disordered regions, things where the context really matters much more than in your standard protein things. So there's reasons to be skeptical. So, I think my poor group heard me go on many, many rants. And so, we decided to do something about it. But just to let me, I don't know how much time. Okay, I have enough time. Just so you can see the kind of madness that is being done in the field. What they wanted to do is they wanted to train a transformer. And just like a transformer was a generative model of language, sequence by sequence, or a generative model of proteins, what I'm trying to do is I'm going to try to predict the next amino acid. What they wanted to do is they wanted to make a generative model for what genes and protein or mRNA counts, because what you really measure is the amount of mRNAs. So a cell would be defined by a set of genes that are expressed, which is G1 through GM, and a set of counts, which is how many mRNAs of that gene do I measure. So here I have gene 1 has C1 mRNAs I measure, gene 2 is C2 mRNAs I measure, so on until GM goes to CM. So that's this capital gene, and that's what defines a cell. And so what I want to know is I want to make a generative model, P of G, for telling me how do I generate a measurement that I would see. right? And so what you start off with is I choose some genes. There's some probability of choosing a gene. It's some categorical probability. And then there's a probability of measuring some count given a gene. And then I just multiply this together. So it's just Bayes' rule. So they're just doing these things. And what they do is they say, okay, I'm going to basically parameterize the probability of this of choosing a gene by some categorical probability distribution, which is just a softmax. And then this pi j is going to come out of a transformer-based architecture. And the counts are going to be some, it doesn't matter, zero truncated Poisson, but it's just something that draws an integer that looks like a Poisson distribution. And then again, this lambda j is going to come out of a large language model, right? And some loss, I'm going to do some loss function that maximizes the probability of seeing the real genes I see, and then matches the counts as close as I can. So it's almost a very vanilla, standard generative model for how these things work. And I'll save you all this stuff, but it's extremely vanilla. 12 layers. The only thing you should take out is that the things we care about are these vectors I get out at the end. So I get a vector for every gene, and I get a vector for a cell, which is just the sum of the vectors for all the genes that are being expressed, right? And the claim is that these vector embeddings tell you something about what's going on in biology in a way normal things couldn't. So the kind of thing they like to say, and I'm going to show you some downstream tasks, which are like predicting cell types across species or classifying cell types. So the kind of thing they're very excited about is that they go to another data set, which is a testes tissue. So there's 8 kinds of testes cells, and they get them from these animals they've never trained on, which are humans, chimp, bonobos. A lot of these are not in the data set that they put in. And then what they say is, okay, I take the vector embeddings for a cell type in, say, Mackay, and then I predict which cell type it is in marmoset. And what I do is I, calculate the embedding for each cell, and then I do a k-nearest neighbor, right? So they call this cross-species training. And what you can see is this thing looks fairly diagonal. So it seems like you can use marmoset data through these embeddings to tell you about Mackay. Or Mackay data to predict about marmoset data. So my question always is how impressed should I be? Is this hard? Is this easy? I think people don't have Bayes estimates for all these tasks. Another thing you can imagine doing is this is the same thing across all pairs of species. And you get some numbers, but again, I have no idea if I should be impressed or if I should not be impressed. The press releases all say this is the most impressive thing on earth. Then another thing they can do is they take all 120 human cell types, and then they say, look how, and they ask how well can I classify these 120 cell types, and what they say is, look, we do really good. And then they compare it to other large language models and they say, this is really good. And they say they do slightly better. So, this is state of the art. So the question again becomes, how impressed should I be? Yeah.
How are they kind of measuring?
The cell types are, the cell types are in standard atlases. All right. So they're defined in atlases by, usually the way they're usually done is that people take a bunch of single cell RNA-seq data. They have some marker genes they know are associated from some biologist told them, these are the genes that define this cell type. Then they make a UMAP, they cluster, they see if those genes are expressed in that cluster, and they call that cluster that cell type. But it's basically, you can roughly say it's like some biological domain knowledge. So these are very well curated. The Chan Zuckerberg has spent a large amount of money curating this data set that they're now trying to exploit with these large language models. So they call this cell by X by gene. So they spent a lot of money trying to standardize and curate these data sets. A lot of money, on the order of $50, $100 million kind of money, So it's hand curated, all right? But the thing that I think, the lesson I teach whenever I teach machine learning is the first thing you need to have is a Bayes estimate of how how hard is the problem in the first place? So what I'm going to show you is that Juan, who's sitting over there, with some thinking, a parameter-free model, so literally 0 free parameters, as opposed to a 1 billion, and a laptop can, in 5 minutes, can actually do better than all the stuff I showed you that the CZI did. So what's interesting is, again, the main thing we have to deal with the fact is that, it was said over and over again that more data is better, but only if you know what the signal in that data is and what the noise in that data is. So it turns out that RNA-seq data, for various reasons in biology I won't tell you about, has lots and lots of variability. And what we found when we plotted the data across cells is that the absolute number of transcripts was quite variable across cells of the same type, but the relative order of things was not changed. So like if something was a 98th percentile in expression in a cell type, it would stay 98th percentile expression, but the absolute number associated with that could change quite a bit. So this suggested, and the second thing we found is that if you averaged all these cell types that were the same, you had pretty well-defined directions in this high-dimensional feature space, this 20,000 dimensional space that corresponded to different cell types. And so this suggested that what we should do is we should just not do any fancy embeddings, but we should just normalize properly and exploit the properties of high-dimensional vector spaces, that basically vectors in high dimensions, if properly normalized, are just perpendicular to each other, right? And so what we did is that we said, what we're going to do is we're going to take each cell type, and we're just going to take the gene and assign it a z-score that corresponds to its percentile. So something at 50th percentile got 0, something at 98.6 percentile got 2, something at whatever the opposite, minus 2 sigma got whatever number it was. So now you 0 standardize all your data, and you just express it in percentile. So that's all we did. And this is something that we figured out for a part of the talk I won't get to, but where, and we have some package that uses linear algebra and these kind of normalization tricks to do, to do stuff that my old student Maria wrote. But the key is to normalize each cell independently and say that all that matters is relative expression. And after you do that, what you can do is you can basically use 2 downstream pipelines. The first is that we basically say, look, we know that we have expressions in a high dimensional space, say gene 1, gene 2, gene 3. We know that the cell types I care about correspond to specific vectors in this direction. And all I can do is I can say these cell types, there's always less cell types than genes. So what I can do is that cell type forms a plane in this high dimensional gene space, so I can just project. Right? So it's just non-orthogonal projection, right? The only thing even remotely subtle about this is that these directions are highly correlated, so you have to use a non-orthogonal projector. Right? So this is just linear algebra and normalization. The second thing I can do, and this is what's this is necessary because this data is so full of noise, is that I can normalize, right? So the data they happen to use has 60,000 genes. I can choose 2,000 of the most highly variable genes, then renormalize, then PCA. So this is just basically throwing out noise while keeping normalization, and then use k-nearest neighbors to classify cell type. Right? So there's no free parameters in any of this stuff, right? I just want to point out there's no, and this is essentially a PCA after normalization and throwing out stuff that doesn't vary. And so we can compare these simple parameter-free pipelines to this, TranscriptFormer. And as you can see, this, I don't know, you don't have to see the numbers, but you can see that there's a lot more blue randomly scattered here, and this is a lot more blue in the diagonal, which is a little embarrassing considering that's a 1 billion parameter model and this has 0, right?
Keep it simple, stupid. Right?
Yeah, exactly. Keep it simple, stupid, right? So that's one result. You can go back to this, cross-species example. So these are 8 species, 8 other species. This is the large language model, and this is linear algebra. And red is good. Yeah, you can now, average this data. The yellow is linear algebra. All the other ones are large language models. These are some F1 scores. I don't even know what they mean, but that's the standard in the field. Now you can try to classify 120 cell types. And here, the problem is that as you add more cell types, you're adding more and more noise. So if you do this k-nearest neighbor pipeline I told you about, we don't quite beat the large language models, though we could if we gave ourselves a few free parameters. But you see, you do nearly as well without doing anything. Right? And so, and so I guess that's it. And what I wanted to just say is that for the small number of types, this linear algebra thing works well. But so what this is showing you is the same score, which, just to give you an estimate, 0.9 is what this scTransformer put out. We got 0.873. This is for 120 cell types. And what we wanted to point out is the reason this is all hidden is actually the most important thing is just the normalization. So if I normalize each cell to itself, I know what the signal is and what the noise is, then even for 20 cell types, I basically get the same performance. It's just that as I add more and more cell types, I'm adding lots of noise, so I have to put an extra PCA in, essentially. I just have to PCA stuff. So I don't know. So it just, more data is not always better if it's noise. The other thing is that before you throw a transformer at something, maybe you should think about things a little bit. And I don't even know what to say. CZI gives me a lot of money. I like them. I like Steve. I like these people. But I think there is a lesson here, which is that they're really good at stuff, but they're not good at everything. And sometimes we should perhaps think a little bit. This is the first time I've given this talk because Juan was just doing this in the last 3 weeks. So I have to figure out how to say this in a little softer way the next time I do this. Hopefully this is not recorded somewhere. But, oh, gosh darn it. But I mean, on some deep level, I always science is going to self-correct itself. But at some level, you realize the self-correction is you, right? So it's kind of interesting, right? So I guess the conclusion is that LLMs seem to be great at capturing structure of DNA sequences due to evolution and demography. At least that's my take on it. Transcription is too noisy for LLMs to represent relationship well. And the biology of cell identity is probably not that well suited for LLMs or large statistical models. And I think, thinking about what the biology is or what the generative process for generating things really matters. And I think that was clear in the math talk too. All the shoulder shrugs was this generative process seems to capture the data much more than other stuff. And so I think large language models seem magical, but I think they're very well suited to capturing certain kind of generative processes of sequences and probably less well suited for other kinds of things. And we should think a little bit. So I think I'll end there because it's the end of the thing.